Hello everyone, welcome back to my channel. Today is the second lecture of our course, that is the overview of the biochemistry, basic to advanced. Today's topic is the biomolecules. In the last lecture, we have already discussed that there are four types of the biomolecules: carbohydrates, protein, fat, lipids, and nucleic acid. Today's topic about carbohydrates. We will cover the carbohydrates in three lectures. Today is the part 1. So let's start today's topic. What is carbohydrate? Carbohydrates are the polyhydroxyaldehydes or ketones and classified into the mono, di, oligo and polysaccharides. The carbohydrates in the di, oligo and the polysaccharides are held together by the glycosidic bonds. The carbohydrates supply the energy and serve as the structural constituents of life. They are primarily composed of the carbon, hydrogen and oxygen. And the most important thing that in the carbohydrate, the hydrogen and the oxygen ratio is 2:1 as in water, like H2O. So hence the carbohydrate known as the carbohydrate. Some of the carbohydrate possesses the empirical formula CH2O whole N, and where N equal to less than equal to 3, satisfying that these carbohydrates are in fact of the carbohydrates. But there are several non-carbohydrate compounds also, like the acetic acid, lactic acid, which also appear as the hydrate of carbon, because we can see that acetic acid, the formula is CH3COOH. That means C2H4 and O2. So here also the hydrogen is to oxygen ratio is 2:1. Similarly, in the case of lactic acid also, it is the non-carbohydrate molecule, but here the number of hydrogen is 1, 2, 3 and 3 here, so total 6, and oxygen molecule is 3. So here also that hydrogen is to oxygen ratio 2:1. And also some genuine carbohydrates are also exist, like that rafnohexose or deoxyribose. They don't have the H is to oxygen ratio 2:1. So generally we can say that carbohydrates cannot be always considered as the hydrates of the carbon. We can define the carbohydrates means it consists of the polyhydroxyaldehyde or ketones or the compounds which produce them on hydrolysis. So what is the function of the carbohydrates? Carbohydrates actually serve as a wide range of the biological function. They provide the energy to playing the structural and the communication roles in the body. Their importance span across all domains of the life. So first function, we can say that it is the main source of the energy in our body. It is a primarily fuel because glucose is the most common and essential energy source for the organism. 1 gram of carbohydrate can give 4 kilocalorie of the energy, and the glucose is actually undergoes the different metabolism process, like the glycolysis, citric acid cycle, oxidative phosphorylation to produce the ATP, and ultimately this ATP is the energy source or the powerhouse of our life. Then it also acts as energy storage, like in the plant body the carbohydrate can store as polysaccharides, and in the animal body the carbohydrate can store as a glycogen, in especially the liver and the muscle. So these forms are the compact, insoluble and easily mobilized also. Next, it can also help as a structural function, like the cellulose, chitin and peptidoglycan. Cellulose actually provides the plant cell wall, the rigidity of the plant cell wall. The chitin, it is a structural carbohydrate in the fungal cell wall and the exoskeleton of the arthropods. Peptide or lichen, it forms the bacterial cell wall. It is the complex form of the carbohydrate and the protein. It can also help as a cell communication and the recognition, like the glycoproteins and the glycolipids actually act as a cell communicator, because the glycoprotein and the glycolipid is the complex form of the carbohydrate and the protein and the lipid, and they act as the molecular signature. They play the crucial role in the immune response, hormone recognition, and also the blood group determination. Next, it is also helpful for the digestive and metabolic health, because the dietary fiber, that means the non-digestible carbohydrates like the cellulose, they actually helped for the bowel movement, regulate the blood sugar level, and also lowers the cholesterol level. Next, it is also help for the development role in the body function, like it is also involved in the fertilization, development of the signaling pathway and the brain function, because the glucose is the explosive fuel for the neurons under the normal conditions. Then it is also helpful for the industrial and the clinical use. It is used in the fermentation industry, because maltose is using in the brewing. The diagnostic tools, glucose strips, we can use for the diabetes monitoring, and also the carbohydrate is useful as a biodegradable material, because now we are producing the starch based plastics also. Next, so we can now describe about the carbohydrates total structure. Already we described that the carbohydrates are the polyhydroxyaldehyde and ketones of the appropriate composition of CnH2ON. So that are components in the biological system. So what is the meaning of these polyhydroxyaldehyde or ketones? So we can say that it is the hydroxyaldehyde group and it is the ketone group. So carbohydrate is the consist of this aldehyde and ketones. So let's describe this structure. In this structure, it is the structure of the glucose. We can say here that in the structure of glucose, the aldehyde group is present. It is the structure of fructose, and in this structure, the ketone group is present. So aldehyde and ketone is present in the carbohydrate. What do you mean by the polyhydroxy? You can say here the multiple OH, that hydroxyl group is present. So that's why we can say it is the polyhydroxy. So that carbohydrates are the polyhydroxyaldehyde or polyhydroxy ketone. So if it is the polyhydroxyaldehyde, then we can say it is the aldose, and if it is the polyhydroxy ketone, we can say it is the ketose. We can classify the carbohydrates in different form also. We can often say the carbohydrate as saccharides. The Greek word saccharon means the sugar. So according to the number of the saccharide in the molecule, we can classify the carbohydrates into the four categories. That is the monosaccharide, the disaccharides, oligosaccharides, and polysaccharides. Monosaccharide means, mono means one. That means one carbohydrates or one saccharide molecule is present here. Disaccharide, the di means two. That means here the two saccharide molecules are present. Oligosaccharide means the number of the carbohydrate or saccharide molecules from 3 to 10, and polysaccharide means more than 10. That is, n number of saccharide or carbohydrate if present, then it is known as the polysaccharides. So let's discuss about the monosaccharide. Monosaccharide is the simplest carbohydrates. Monosaccharide are the basic units of the carbohydrate. They cannot be hydrolyzed into the simple sugar and serve as the building block of disaccharide, oligosaccharide or polysaccharides. The empirical formula of the monosaccharide is CH2O whole N, and N is 3 to 7 here. Monosaccharides are either the straight chain or the cyclic forms. The detailed structure of the monosaccharide we will describe in the next lecture. Next is taste. The carbohydrate taste is sweet. Solubility, it is highly soluble in the water. And in carbohydrate, in the monosaccharide, two main functional groups are present, either aldehyde or ketone as carbonyl group and multiple hydroxyl group. If in the monosaccharide, the aldehyde is present, like in the glucose and the galactose. It is known as the aldose. And if the ketone group is present, like the fructose, it is known as ketose. So it is the structure of the glucose. So we can say here multiple OH group is present and the aldehyde group is present. So it is under the aldose family. And in the case of fructose structure, here the ketone group is present and multiple hydroxyl group is also present. So it is the polyhydroxy ketone and it is also known as the ketose family. The classification of the monosaccharide are based on the number of carbon. That we already told that in the monosaccharide the N should be 3 to 7. So according to the number we can classify it into the different sections like the triose family, tetrose family, pentose, hexose and heptose. So if the number of carbon is 3, it is under the triose family. If the number of carbon is 4, then tetrose. Simultaneously, if it is the 5, then pentose, 6, hexose and 7, heptose. So each category again we can classify into the two parts. One is aldose and another one is the ketose. So under the aldose, the triose family member is glyceraldehyde. Under the ketose, the triose family member is the dihydroxyacetone. Similarly, in the tetrose family, it is the erythrose and erythrulose. Under the pentose, it is the ribose and ribulose respectively. For the hexose, it is the glucose and fructose. And for the heptose, it is the glucoheptose and pseudoheptulose. Next, the properties. So one of the most important property for the monosaccharide is the isomerism. There are different types of the stereoisomer. So first we describe what is stereoisomer. Stereoisomers are the compound with the same molecular formula and same sequence of bonded atoms that differ in their three-dimensional orientation for their atoms. What is stereo center? The stereo center means it is the chiral center. That means it is a carbon that is bonded to 4 different atoms or group. This carbon is also known as the chiral carbon. So according to that, we can classify different type of stereoisomer in the monosaccharide. So first is the D and L configuration. It is under especially under the Fischer projection. The detailed structure of the monosaccharide in the Fischer projection, the Haworth projection, the Cheer conformation, everything we will describe in the next lecture. So the system is based on the orientation of the hydroxyl group on the chiral carbon farthest from the carbonyl group, that is the reference carbon. This is actually the definition of the D and L configuration. Again, I am saying that D and L configuration, it is based on the orientation of the hydroxyl group on the chiral carbon farthest from the carbonyl group. It is the structure of the one general structure of the carbohydrate. Here this 1, 2, 3, 4, 5, 6 all are the chiral carbon, all because we can say, we already told chiral carbon means the carbon which actually bonded to 4 different atoms. So let's we describe this is a chiral carbon. So one hand hydrogen, one hand OH, one hand CH2OH and another hand this group. So 4 different groups are there. So this is the chiral carbon. So it is the orientation of the hydroxyl group of the chiral carbon farthest from the carbonyl group, that is the reference carbon. So with respect to this CHO group, the farthest chiral carbon is the C5, and the orientation of the hydroxyl group of this C5 actually determines that it is D configuration or it should be the L configuration. Similarly, in the case of the ketose family, the farthest distance from the ketone group, that is the again this C5, it will again the position of this hydroxyl group in the C5 position will describe it is the under the D configuration or L configuration. We can say that if the OH is in the right hand side, if the farthest chiral carbon from the reference carbonyl group, the OH is in the right hand side, then it is under the D isomer. If OH is in the left hand side, then it is the L isomer. The same will follow in the case of the ketose family. If the OH is in the C5 carbon, that means the farthest chiral carbon from the carbonyl group, if it is in the right hand side, then it is the D isomer. If it is in the left hand side, then it is under the L isomer. So the structure overview, that we can say that carbon chain is drawn vertically like this, and with the most oxidized group like the aldehyde or the ketone at the top, and each intersection represent a chiral carbon. Each intersection, this 2, 3, 4, 5, all the intersection are the chiral carbon, and these horizontal lines point towards the viewer and the vertical lines point away. Why this D and L configuration is too much important? Because in our body, all the metabolic function is carried out by the enzymes, and the enzymes are stereospecific, meaning they usually recognize either D forms and L forms, but in the case of carbohydrate, they are mostly recognizing the D form. So that's why it is very necessary to understand that what is the D configuration and what is the L configuration. We already described that D form means that the OH group in the chiral carbon from the farthest position from the carbonyl group, it should be in the right hand side. Then it is the D form. And if the OH group in the left hand side, then it is the L form. So the D form is biologically active form for most of the naturally occurring sugar, and the L form is actually the mirror image of the D form and generally not metabolized by the human enzyme. Then what is the enantiomer? Enantiomer means we can say it is the mirror image isomers. Enantiomers are a pair of stereoisomers that are non-superimposable mirror image of each other, just like our left and right hand. We can say that D form and L form, if we can keep side by side, we can say that D form and L form are the mirror image of each other. So the D form and the L form of one molecule is the enantiomer. They differ in the spatial arrangement around one or more chiral carbon. Each enantiomer has opposite configuration like D versus L, but they have the same molecular formula and same sequence of the bonded atom. Just it is the mirror image. You can say that in this D glucose, the H is in the left hand side, OH in the right hand side. Similarly, in the L glucose, OH in the left hand side, H in the right hand side. So this is just the mirror image relation. Next, the epimers. The epimers define, it is the stereoisomers that differ in configuration at only one specific chiral carbon. at only one specific chiral carbon other than the anomeric carbon. Among two or more stereocenter. We already described the stereocenter means it is the chiral center. Each chiral center we can say it is the stereocenter. They are not mirror image like the enantiomer. So what we can say that if we consider that glucose and galactose, so all the structure are same except this C4. In the C4, the D-glucose, H is in the left hand side and OH in the right hand side. But in the D-galactose, OH in the left hand side and H in the right hand side. So we told that epimers means they differ in the configuration at only one specific chiral carbon. So in the glucose and the galactose structure, only they are differentiating in the C4 chiral carbon. So D-glucose and D-galactose are the epimer of each other at C4. Similarly, if we consider the D-mannose and D-glucose, we can say all the structure are same except this C2. So we can say D-mannose and D-glucose are again epimers at respect to C2. But the mannose and the galactose are not the epimers. Next, the diastereomer. Diastereomers are the non-mirror image stereoisomer. Diastereomers are a type of stereoisomer that are non-identical, non-mirror image of each other and non-superimposable also. They have same molecular formula and the bond connectivity but differ in the spatial arrangement of atoms at one or more chiral center. They occur when two or more stereoisomers of a compound have different configuration at one or more of the equivalent stereocenter and are not mirror image of each other. We can say epimers are the subcategory of the diastereomer. Like we already explained, the D-glucose and D-galactose are the epimers of each other at C4. We explained that D-mannose and D-glucose are the epimers of each other at C2. But D-mannose and D-galactose are not epimers of each other because they are differentiating in C2 and also C4. But these D-mannose and D-galactose are diastereomer of each other because if more than one chiral carbon is different, then we can say they are the diastereomer and also they are non-identical, they are non-mirror image of each other and also they are non-superimposable. So that's why we can also say that epimers is a subcategory of the diastereomer. Then what is the anomer? Anomers are a type of stereoisomer, a subtype of epimer also that differ only at the anomeric carbon. The carbon derived from the carbonyl carbon, that means from where the aldehyde group are belonging, from where the keto group is there, this particular carbon is known as the anomeric carbon. So if the configuration is differ only at the anomeric carbon, then we can say they are the anomer of each other. In the aldose, the C1 where the aldehyde group is there, it is the anomeric carbon. In the ketose, the C2, it is the anomeric carbon. Then alpha and beta anomers. Actually anomers are two types. One is alpha anomer and another one is the beta anomer. Alpha anomer means if the OH group on the anomeric carbon is trans with the CH2OH, then we can say it is the alpha anomer. And if the OH group is cis to the CH2OH group, then it can say as the beta anomer. This projection is known as the Haworth projection. in the carbohydrate, three types of projection is there. One is Fischer projection, one is Haworth projection, and one is chair projection. We will describe all the structure in the next topic. So, if the OH and CH2OH is in the trans orientation, then it is known as the alpha anomer. If OH and CH2OH is in the same site, then it is known as the beta anomer. We can also say that it is the alpha anomer and it is the beta anomer and it is the open chain form. Then what is about the muter rotation? Muter rotation, we can say it is the interconversion of anomer. We already explained that there are two types of anomers are there, alpha anomer and beta anomer. So the interconversion of this alpha and beta is under this muter rotation. Muter rotation is the spontaneous change in the optical rotation of a sugar solution due to the interconversion between its alpha and beta anomeric form through the open chain structure. How it happens? It is a sugar like D-glucose dissolved in water. Its cyclic alpha and beta form opens temporarily to form a linear or open chain structure. Then this linear form recloses and they form either the alpha form or the beta form. An equilibrium mixture is established between all these three forms. This mechanism is known as the muter rotation. So in this mechanism we can say that alpha and beta D-glucose exhibit in the muter rotation form. If any form is dissolved in water, let's the alpha. If it is dissolved in the water, the specific rotation in the case of alpha D-glucose decreases from the 112 degree. Normally, alpha D-glucose has a specific rotation value of 112 degree. Similarly, beta D-glucose have a value of 18.7 degree. So, if the alpha D-glucose is dissolved in the water, then the specific rotation value will decrease from this 112 degree, and if the beta D-glucose is dissolved, then the value is increased from 18.7 degree to the value of 52.7 degree. So this phenomena is called the muter rotation because in this 52.7 degree, the glucose is exist as the open chain structure, alpha D form and the beta D form. And in the equilibrium mixture, the alpha D form percentage is 36 degree, 36%, beta D glucose percentage is 63%, and open chain structure percentage is 1%. Combinedly, they exist as an equilibrium mixture has a specific rotation value of 52.7 degree. What is optical isomerism and racemic mixture? What is optical isomerism? It is a type of stereoisomerism when the molecules have the same molecular formula and same bond connectivity but differ in the three-dimensional arrangement of atoms in space, especially how they interact with the plane polarized light. Optical activity means when a beam of plane polarized light passes through a solution of the chiral compound, the orientation of the light is rotated either to the right direction or to the left direction. This property is known as the optical activity. And the molecules which exhibit this behavior is known as the optical active compound. What is the meaning of plane polarized light? The light that vibrates in only one direction. And optically active compound that rotate this plane of vibration. The types of optical isomerism. There are two types. One is the dextrorotatory isomer which can also known as the plus form or D form, and levorotatory isomer, the minus or L form. Dextrorotatory, that rotates the plane polarized light to the right side, that means the clockwise direction. It is designated as plus sign or D because the Latin word dexter means right. Example is D-glucose. Similarly, the levorotatory isomer, that is the minus or L form, it rotates the plane polarized light to the left because the Latin word the levers means the left, that means counterclockwise. It is designated as minus or small l. The most important thing, it should be remembered that capital D and capital L configuration system is totally based on the structure. That means the orientation of the hydroxyl group of the chiral carbon farthest from the carbonyl group. If it is in the right hand side, then D. If it is in the left hand side, then L. This capital D and capital L system is not always the same as the plus or minus system or small d or small l. They may or may not correlate. What is racemic mixture? Racemic mixture means if contains the equal amount of this dextrorotatory and levorotatory isomer, then it is known as the racemic mixture. The optical activity cancel each other, so the net optical rotation is 0. That small d, the dextrorotatory isomer has a optical activity, and levorotatory isomer has also a optical activity. But if equal amounts of the dextrorotatory and levorotatory isomer will present, then the final outcome of the mixture, the optical activity is 0. So the racemic mixture are optically inactive, even though the individual isomers are active. Like we can say the racemic mixture, that optical active 50% D isomer and optically active 50% L isomer make that optically inactive racemic mixture. Next, like the example of DL glucose, that means the racemic mixture of D glucose and L glucose. So if it presents the equal amount of D, that means plus glucose and minus glucose 50-50%, then it will make the DL glucose the racemic mixture, and the racemic mixture of this DL glucose is optically inactive. D-glucose is dextrorotatory, rotate the plane polarized light to the right direction. L-glucose is levorotatory, it will rotate to the left direction, that means counterclockwise. But when combined in equal concentration, that 50% D and 50% L, the optically effect cancel out. The mixture becomes optically inactive. So it is the racemic mixture. But DL glucose is rare in nature because naturally occurring glucose is almost exclusively D glucose, known as dextrose. L glucose is rarely found in the nature and not metabolized by most of the enzyme in human body. So DL glucose, the racemic mixture of this glucose, usually made synthetically in the laboratory. The next property is the reducing property of the monosaccharides. All the monosaccharides are the reducing in nature. Reducing sugar means it is the carbohydrate that can donate electrons to another chemical, typically by the reducing metal like the copper plus 2 or the silver plus, due to the presence of a free aldehyde or keto group in its structure. The aldehyde group of the glucose is oxidized to a carboxylate group, that RCHO. it will convert it to RCOO minus. So aldehyde group is converted to the carboxylate group. And the copper or Ag plus ions in the reagent accept these two electrons releases by the glucose and get reduced to copper plus, which precipitate as Cu2O. Similarly, the silver ion, it can also accept the two electron which is removed by the aldehyde of the glucose molecule, and it will form the silver and precipitate it. So the glucose is oxidized because it loses two electrons, and the copper plus or silver plus ion is accepting these two electrons, so it is actually reduced. So these electron transfer validates that glucose act as a reducing sugar because it is donating electron. It is act as a reducing sugar. It is oxidized, and the copper plus 2 or silver plus, which is present as a chemical agent, the reagent, it is gains these electrons. So we can say that overall reaction, the RCHO, if it is the glucose or whatever any monosaccharide, it is plus the copper plus 2, which is from the reagent, and it is actually converted to the carboxylate group, that RCOO minus, and 2 electron it is removing. These 2 electron, this copper plus 2 is accepting and form the Cu2O, which is precipitate, and water is generated. So why all monosaccharides are reducing in nature? All monosaccharide contains the free aldehyde or ketone groups. For the aldose family, it is exists as a free aldehyde. For the ketose family, it exists as a ketone group. The aldoses have a free aldehyde group in the open chain form. Ketoses, although they lack a direct aldehyde group, but they can isomerize to aldose in the alkaline solution, thus also act as a reducing sugar. Reducing sugar can reduce the mild oxidizing agent like the Benedict, Fehling, Tollens because of the presence of this free aldehyde and ketone group. Because this free aldehyde and ketone group actually releasing the electron, and the mild oxidizing agent is accepting those electrons. In aqueous solution, especially in the alkaline medium, the monosaccharides interconvert between the cyclic and open chain form. When it is exist as the open chain form, then the aldehyde and the keto group is free, and at that time this free aldehyde and keto group is participating in this oxidation reduction reaction. So in the case of the Benedict test, it is a qualitative chemical test used to detect the presence of the reducing sugar in a solution. That means the presence of the aldehyde and keto group, we can use this test. We can perform this test. What is Benedict reagent? It is the composition of the copper sulfate, which is the source of this copper plus 2 ion, the sodium carbonate, which provides the alkaline medium, and the sodium citrate, which actually stabilize the copper plus 2 in the solution. So the mixture of the copper sulfate pentahydrate, the sodium carbonate and sodium citrate, the mixture is known as the Benedict reagent. What is the procedure for this test? Add 2 ml of Benedict reagent to the test solution. Heat the mixture in the boiling water bath for 2 to 5 minutes. Observe the color change, that you can get a brick red precipitate of Cu2O. But all time the brick red precipitate is not appeared. Sometimes green, dark green, yellow, orange, and precipitate can also come depending on the percentage of the reducing sugar in the sample. So if the concentration of the reducing sugar in the sample is maximum, then the brick red color will appear. What is the overall reaction? Overall reaction is CH2 plus copper plus 2 from this Benedict reagent. OH minus means it is the alkaline solution. So the aldehyde group is converted to the carboxylate group. The copper plus 2 is converted to the Cu2O. So the red color precipitate will come and water will generate. So the oxidation of sugar, if we what is the oxidation reaction and reduction reaction if we describe. So the free aldehyde group from the glucose or any aldose is oxidized to a carboxylate group RCHO to RCOO minus, and then 2 electron is generated. These 2 electron will accepted by the copper plus 2, which can derive from the Benedict reagent, and it will convert it to the copper plus. Then the copper plus combine with the hydroxide ion from the alkaline medium to insoluble copper oxide, and this Cu2O is giving the brick red precipitate. Depending on the percentage of the reducing sugar in the sample, the color may vary. The same type of reaction can also happen in the case of the Felling's test. The Felling's test is similar like the Benedict test. It can also detect the presence of the reducing sugar in a solution. What are the reagent composition for the Felling's reagent? That is the copper sulfate, source of the copper plus 2 ion, the sodium hydroxide, which provides the alkaline medium, and the potassium sodium tartrate, that is the Rochelle salt. It is the complex Cu plus 2 ions. So the copper sulfate solution is the Felling's A solution, and the alkaline solution of this potassium sodium tartrate is known as the Felling B solution. So if we mix this Felling's A and Felling B solution, ultimately the Felling solution we will get. So every time at the time of experiment, we have to prepare the freshly Felling solution. Felling's A and Felling's B, it is available in the market, but we have to mix to prepare the final Felling solution. So add 2 ml of the Felling solution to the test solution. Heat the mixture in boiling water bath for 2 to 3 minutes. Observe the color change. Again, the red color precipitate will come. Brick red precipitate of the copper oxide. Overall reaction is same as it is the Benedict test. That the aldehyde group from the aldose, it will convert it to the carboxylate group and it will remove 2 electrons. So it is the oxidation reaction. These 2 electrons will accept by the copper plus 2 ion, then it will form the copper plus, and in the alkaline solution, in presence of alkaline solution, this copper plus will convert it to the copper oxide, and it is give the precipitate of the brick red color. And so here the glucose is act as a glucose, or any monosaccharide, any aldose family or ketose family act as a reducing sugar. Next, the tolerance test. The tolerance test is a qualitative chemical test used to detect the presence of the reducing sugar, particularly those containing the aldehyde group. So we can distinguish between the aldose and ketose, and also identify the reducing sugar in the solution by the tolerance test, because the Benedict test and the Felling's test both will give the positive result for the aldose and ketose. So if we try to further distinguish between the aldose and ketose, then we can try with the tolerance test. What is the reagent composition for the tolerance test? The silver nitrate, which is the source for the silver plus ions, and ammonium hydroxide added dropwise to the precipitate Ag2O, and then dissolve it. So it is forming the diamine silver complex. What is the procedure? Prepare the tolerance reagent freshly before the use. Add 2 ml of tolerance reagent to the test solution in a clean test tube. Heat the mixture gently in a warm water bath. observe the result after 2 to 3 minutes. So we can get this type of result, that silver precipitate it will come, that mirror like image, the mirror like precipitate will form. So silver mirror on the test tube. If the percentage of the aldose group is less, then some gray color, some PPT will appear. What is the overall reaction? That carbohydrate, any type of aldose molecule, and this diamine silver complex, it is the alkaline solution. So ultimately these aldose will converted to the carboxylate group. Diamine silver complex will take the 2 electron from the aldose and it will converted to the silver precipitate, and the ultimate reaction, the ammonia and water will generate. So what is the non-reducing sugar? The non-reducing sugar is a carbohydrate that does not have a free aldehyde or ketone group, therefore cannot reduce the mild oxidizing agent like the Benedict, Fellings or Tollens reagent. All the monosaccharides are the reducing sugar, and the disaccharides and the polysaccharides are the non-reducing sugar. The sucrose, the disaccharides, is the non-reducing, and all the polysaccharides are the non-reducing, because in the case of polysaccharide, all the carbohydrate, all the carbohydrate, the aldose or the ketose group is not free. It is the blocked by the glycosidic linkage. In the non-reducing sugar, the aromatic carbon of both the monosaccharide units are involved in the glycosidic bond, glycosidic linkage. So it prevents the sugar from converting into a open chain form with a free aldehyde or the free keto group, which is necessary for the reduction. So in the case of the polysaccharide, it is not free. So that's why all the polysaccharides are non-reducing sugar. And in the case of disaccharide, only the sucrose is non-reducing. So, from this today's lecture, what is the key points? That is, what is the definition and the composition of the carbohydrate? That carbohydrates are the polyhydroxyaldehydes or ketones that primarily composed of the carbon, hydrogen and oxygen. And the most important thing, that here the hydrogen is to oxygen ratio is 2:1. So the general formula CH2O whole N. But if in the carbohydrate formula the hydrogen is to oxygen ratio is 2:1, then we can say this carbohydrate is a carbon hydrate. But many carbohydrates molecule are there which is not satisfying the H:O ratio 2:1. So we can generally say that carbohydrates means it is the polyhydroxyaldehydes or ketones, or a compound which after hydrolysis it will give the polyhydroxyaldehyde or ketones. The classification, we can categorize the carbohydrate into the monosaccharide, disaccharide, oligosaccharide and polysaccharide based on the number of the sugar units. If one saccharides or carbohydrate, then mono, if 2, di, if oligo 3 to 10, and if more than 10 or n numbers, then it is the polysaccharide. There are different function of the carbohydrate, like it is the main source of energy. It can also store the energy like the starch and glycogen, respectively in the case of plants and animals. It can also give the structure like the cellulose for the plant cell, chitin for the fungal, peptidoglycan for the bacterial cell. It can also help for the cell signaling through the glycoproteins and the glycolipids. It can also give some health benefit like the blood sugar regulation, dietary fiber, cholesterol level regulation, etc. It can also use in the industrial purpose like the fermentation, bioremediation, diagnostics, etc. Then there are different types of isomerism in the case of monosaccharide. Monosaccharide as the simple unit, it is highly soluble, sweet in nature. They can form the capital D and capital L isomerism. enantiomer, epimer, anomer, mutarotation. The optical activity is also possible for the case of the monosaccharide. So small d and small l form, that is the dextrorotatory and levorotatory structure is also possible. Racemic mixture contain the equal D and L form and make the mixture is optically inactive, though the components, that is the D and L form, is the optically active. Reducing it, all the monosaccharide have a reducing property. All the monosaccharide acts as a reducing sugar because all the monosaccharides have a free aldehyde and the keto groups. So they can oxidize to the carboxylate and reduce the mild oxidizing agent like the Benedict Fellings or the toluenes to the Cu2O or the silver. So by this test we can identify the monosaccharide or we can identify the carbonyl group of the monosaccharides. That Benedict and Fellings are same. They are giving the positive result for the aldehyde and the keto group both, but if we can if we try to distinguish between the aldose and ketos, we can try for the toluenes test, because toluenes test is giving only the positive result for the aldose, and all the non-reducing sugar, that means which don't have this free aldehyde and the free keto group, they are under the non-reducing, and all the polysaccharides, because they don't have the free carbonyl group, that's why all the polysaccharides are the non-reducing in nature. Disaccharides, the sucrose is also non-reducing in nature. Thank you. We will come back in the next lecture with the detailed structure of the carbohydrate, and in this lecture we will explain about the Fisher projection, Haworth projection, Cheer conformation, everything for the monosaccharides. Also we will describe about the structure of the disaccharides and the polysaccharides. Thank you.